In section 4.1, we start off talking about the role of microscopy. You can't really talk about cells until you talk about microscopes, right? I mean, it wasn't really until the advent of the earliest microscopes that we start to really get a picture that there were these cells. And starting with von Leeuwenhoek and other scientists, Robert Hooke, they're the ones who really advanced our understanding that there is this microscopic world because of the availability of microscopes. Now, before we get into what a microscope does, etc., let's just define a cell. And this is one of the many theories that you'll see in biology. Now, it's very important that we define that. A theory is not a guess. A theory is a hypothesis that's not been disproven by repeated independent experiments. It is our best understanding of how the world works. Evolution is a theory, not a guess, a theory that has not been disproven by a preponderance of the evidence. Cells, there's a theory that applies to cells. All living things have one or more cells. A bacteria is a living organism. It is single-cellular, bacterium. A human is multicellular, also considered a living organism. Cells are the smallest units capable of carrying out the processes of life. We've talked about the fact that cells have a collection of proteins and carbohydrates and lipids and nucleic acids, all of which comprise the cell, all of which function in unison to make a cell. On its own, a protein is not considered living. A lipid is not considered living. It can't react to external stimuli, require energy. These are not living entities. But now when you organize them in a very specific way into what we know to be a cell, the cell is the basic unit of which all life is built. The last and most important part of the cell theory, new cells only come from pre-existing cells via cell division. I can't take fats and carbs and proteins and nucleic acids. I can't just take a bunch of carbon, oxygen, hydrogen, nitrogen. I can't stick it all together and hope a cell forms. I can only get a cell from an existing cell. Now, of course, the first question that comes in, where did the first cell come from? And that is something that's under intense scrutiny. There's a lot of hypotheses. This is something that scientists have been very interested in understanding. But regardless of how the first cell arose, we're going to learn more about how cells arose in the evolution part of the course. But be that as it may, once the first cell arose, all cells came from that existing progenitor. And so if you think about it, all cells, all living things are made of at least one cell. All cells come from pre-existing cells. There's a unity of life here. There was an original cell that evolved, that underwent natural selection, that started to branch out, that then created all the diversity that we see on Earth today. So microscopy. Microscopes are used to observe cells. Now there are three basic parameters. The first one, very straightforward, magnification. It's the ratio between the size of an image produced by a microscope and its actual size. In our lab, we have microscopes that can magnify 40 times the original size, 100 times the original size. We have lenses that can go all the way up to 1,000 times the original size. Now the question is, why don't our light microscopes in the laboratory go up to 2,000 to 10,000? We're limited by the resolution which can be achieved by certain microscopes. Now, resolution is essentially a measure of clarity. The formal definition is it's the ability to observe two adjacent objects as distinct from one another. So 2 points that are in close proximity can be observed as being separate. So the resolution of my eye is enough to tell these are two separate entities. At some point, when these objects become too small, my eye is limited in its resolution. Even though I can see something is there, I can't resolve the image. So the real limitation of a microscope is the ability to resolve an image, not the ability to magnify it. The third thing you need to take into account when using a microscope is how you can create contrast in order to have your object appear different than its background. For example, most cells are colorless. So if you shine white light through that sample, you're going to have a white background and a white cell. It's going to be very difficult for you to be able to make out any details because you lack sufficient contrast. So we often use dyes to add color to a specimen. So now against that white background, your red object stands out. Now there are two types of microscopes that are based on their source of illumination. It's the source, the illuminating source, if you will. You have a light microscope, which uses white light, visible light. The resolution on this is about 0.2 microns. So for objects smaller than 0.2, we're going to be unable to resolve the image, be unable to really make out any clarity of that image. If you want to see something smaller than 0.2, then we resort to an electron microscope, which uses an electron beam to excite the specimen. That resolution is much more powerful, goes all the way down to 2 nanometers. So a micrometer is 10 to the negative 6. A nanometer is 10 to the negative 9. So what we have here is 2 times 10 to the negative 7 meters. And here we have 2 times 10 to the negative 9th meters. So you can see electron microscope. It's about 100 times more powerful from a resolution standpoint than a light microscope. To give you a sense of scale here, of course a human, a bird's egg, a fish egg, all of those are able to be viewed with the unaided eye at about 200 microns. The eye becomes limited. We then can turn to a light microscope to look at plant and animal cells, to look at small single-celled organisms, even large organelles, as you observed, I'm sure, in A&P, can be observed using a light microscope. But again, when you get up to about 0.2 micrometers or 200 nanometers, we become limiting, which prevents us from seeing very small bacteria, viruses, most organelles, and then at the atomic and the molecular level, we're limited, obviously much limited with the light microscope. So the electron microscope then gives us details that we couldn't get for those objects. Notice though, the electron microscope does not have atomic resolution. There are actually now current microscopes that do allow us to get that low on the resolution scale, atomic force microscopes. So what microscope will you choose to use? Well, it depends on how small the object is. For most cells, the light microscope is fine. For particles like viruses, for organelles, intracellular particles like ribosomes, and macromolecules like proteins, we require an electron microscope to make out the details. So there are two types of electron microscopes. You have transmission electron microscopes, which the beam of electrons is transmit through the sample, gives us a sense of the internal organization of a specimen. Notice that thin slices of the specimen are stained with heavy metals. You cannot look at living specimens. You can only look at preserved and dead specimens using electron microscope based on what you have to do to manipulate them. A scanning electron microscope coats the sample with the heavy metal, which then results in the beams of electrons bouncing off the surface. This will give us an idea of the external organization and appearance of a sample. Here's an example, right? An egg being fertilized. Here in a transmission electron micrograph, we can see and observe the inside of the eggs, the internal architecture. We can't really make out much of the surface here as we can with a scanning electron micrograph, where we see actually a sperm attempting to penetrate the outer barrier of that egg. In each case, the increased resolution of each microscope allows us to see details and structures we would be unable to see using a simple light microscope.